All right, hey friends, it's T-Man, and I'm back again with another video, an educational video. Yes, man, because sometimes people ask me, hey, man, you know the stuff that you post in the community section, can you, do a video on it? I'm like, yeah, okay, sure. So this is just one of the things I've posted, recently. I post a lot of stuff like this all the time, man. There's just so much stuff out there. It's incredible. It's incredible that science actually reveals through these studies that they did on people to tell you what builds muscles. Get it? What type of muscles you're getting, how it's built, why it's built, this, that, everything. It's amazing. Science tells you everything. Tells you why you don't get damaged anymore because repeat about effect. Tells you what this force production is based on. Tells you what these phony muscles are based on, sarcoplasmic as opposed to myofibrillar. It is good to know that, don't you think? Going to a gym, lifting a weight, all these young people I talk to on the internet, they're like, hey man, do you know what sarcoplasmic hypertrophy is? And they're like, no, I don't know what that is. It's a good thing to know what you're getting in the gym, right? As opposed to artificial muscle growth and real muscle growth, myofibrillar to sarcoplasmic, isn't it? Don't you think so? Yeah. It's kind of a wake-up call when you find out, oh shit, I just been building fake muscles all this time? Yeah, just for the visual effect of it, the visual effect to look, pretend jacked. Pretend jacked, get it? I just love that one. Pretend jacked, sarcoplasmic, pretend jacked. Okay, the mode of myofibrillar remodeling, this is PubMed. Remodeling of human skeletal muscle affected by DOMS. It's affected by DOMS, bro. Induced by eccentric contractions. They always talk about eccentric. They never really talk about concentric stuff in these studies. I don't know why. I guess concentric sucks. It's just pushing a weight. It's a force produced, a force produced. All right. So muscular Z disc streaming, the loss of the destamine skeletal are considered the morphological hallmarks of eccentric contractions induced injury. Okay. The latter is contradicted by recent studies where a focal increase of desmin was observed in biopsies taken from human muscles with DOMS. Oh, with DOMS. See eccentric contractions and DOMS. In order to determine the effects of eccentric contractions induced alterations of the myofibrillar Z disc, we examined the distribution of alpha, and so they did this thing on DOMS, biopsies, whatever. I'm not going to go through this whole list. This is just a short thing I'm going to actually go elaborate more in another study. This was just one study, okay? The more important is the bottom part here, the conclusion. Okay, these were preferentially seen in regions with increased numbers of sarcomere and parallel myofibrillars. Look, DOMS and eccentric exercises are what? These were preferentially seen in regions with what? Increased numbers of sarcomeres and parallel myofibrillars. Oh, how about that, eh? You're getting more muscle fibers. We propose that these staining patterns represent different stages of sarcomere formation. We want to form sarcomeres, yes. We don't want to just get sarcoplasmic hypertrophy. We don't want to load a lot of glycogen in our muscles to pretend we have phony muscles, to pretend we have muscles, creating phony muscles. So they make you stronger, they make you live longer, and they make you healthier and better and wear off all kinds of disease, real muscle. But if you're not building muscle and you keep putting glycogen in your muscles, you keep loading glycogen, eating an artificial diet called carbohydrates, then it makes you toxic. There's toxic buildup in there. It's unhealthy for you, makes you sick, and will shorten your lifespan, and you'll die eventually. It'll keep you alive probably for a while because it's energy, it's a drug. You're loading like eating carbohydrates. There's drugs. Sugars is a drug. Anyway, so whatever. The findings therefore support our previous suggestions that muscle fibers subjected to eccentric contractions adapt to what? Unaccustomed activity by the addition of new sarcomeres. Oh, so how do muscles adapt? Let's see. How do they adapt? Well, when there's an eccentric contraction, right, to an unaccustomed activity or exercise or sporting event, what happens? There's an addition of sarcomeres, new sarcomeres. What's a sarcomere? It's a spongy material inside, the filaments, whatever, the proteins, contractile. Let's just say contractile proteins, whatever, these real contractile proteins inside. You're getting some real muscle growth there, okay? So yeah. There you go. How about that? It's interesting. There's more stuff down here. There's always more stuff. DOMS. I'll elaborate more now. I'll go into another one. So this one's pretty good too. Yeah. Let's open that one up. Boo. Boo. So muscle regeneration, cellular and molecular events. Well, I got to tell you here, man. It's a short. This one's short too. Muscle injury induces strong changes in muscle cells and extracellular matrix. Oh, muscle injury. Well, what they're talking about muscle injury, they're not talking. There's minor injuries and then there's really bad injuries. This is a minor injury. Okay, dude, you gotta relax. It isn't. I got injured like with a hammer in my head or something. It's a micro injury. Muscle regeneration after. They should say micro injury. Micro injury has similarities to muscle development during embryogenesis and seems to follow the same procedure. The initial phase of muscle repair is characterized by inflammation and degeneration of the damaged muscle tissue. Okay, so degeneration, that's a necrosis, a necrosis or an apoptosis. You could call this an apoptosis or a necrosis. That's what happens when muscles get damaged. There's an inflammatory response and then there's a degeneration. During that degeneration, that means that protein synthesis needs to exceed that degeneration, that breakdown, in order for muscle to build. Let me explain this. If you ever damage muscle fibers in a gym and you get DOMS, like it said there previously, and that DOMS will last 1, 2, maybe 3 days. Sometimes it doesn't last that long. Depends on the amount of damage. If it was really good micro damage, it could last 3 to 4 days. Okay, about 4 days, something like that. If it was really, really minor, it may last a day, maybe 2, maybe 1 day at the most, tops. Then there's not really a lot going on there. But however, protein synthesis still needs to exceed that breakdown. If it doesn't, you'll lose those muscle fibers. They'll die and go away. So you wanna exceed that so you could be in an anabolic growth state so you could repair these muscles. How does being in an anabolic, how does protein synthesis exceeds this breakdown put you in an anabolic growth state? It's really easy. When it exceeds that breakdown, what happens is it shuts off the catabolic hormone. The catabolic hormone converts to an androgen, so it doesn't have to compete with the myocytic androgen receptor. So now you're anabolic. So now these muscles can rebuild. They can regenerate and rebuild bigger and stronger, and you can get more fibers doing that. That's how it works, man. Yeah. Okay, so almost simultaneously, previous quiescent myogenic cells called satellite cells, the progenitor cells, these stem cells, they're activated. They're activated through what? These microinjuries. They proliferate, differentiate, and fuse to form multi-nucleated myofibers. That's what they do. They fuse together and they create what? Fibers. They create new fibers, and these fibers are multi-nucleated. If a fiber is created, it's created with nuclei. They're highly myonucleated. Okay, other non-muscle stem cells may also take part in this process. Yeah, they do. They can. There's mesenchymal cells, a whole bunch of other stuff. They can come out and they dance in this process. Secreted factors such as hepatosate, or whatever. I can't even pronounce these words, man. Growth factor, HGF, fibroblast growth factors. They're released, yes. Protein synthesis is extended. Blood levels are elevated during the repair and the remodeling phase, which takes about a week, and depending how much damage, it could take to 14 days, but mostly it's 7 days. I can do it in 7 days if you're in an anabolic growth state. If you're eating enough, protein synthesis exceeds this breakdown. Transforming growth factor betas, TGF betas, insulin-like growth factors. Well, if you're adding carbohydrates, a good starch like potatoes, well, this won't spike insulin, but the insulin will definitely, increase. And that could help too in this repair and remodeling because, it helps with packing. On your natural diet, which is a carnivore diet, your body will release ketones, but you can really speed up this process if you were to build more muscle fibers. There's more space, so you eat some carbohydrates or starch. Tumor necrosis factor alpha, complex carbohydrate, and others are released during muscle repair and guide muscle regeneration. However, their exact functions and effects on muscle remodeling remain unknown. Intensive research is currently addressing the regenerative mechanisms which are involved in acute muscle injury and chronic muscle diseases. Well, that's separate, because that's just part of the study. They were talking about stuff, regeneration. So cellular molecular regulation of muscle regeneration. Is this regulation? Regulation. There's so much more here. Every time you open this, there's like a million other sections that come out. I don't even know what this one is here. Did I read this one? I think I read this one before. It says more about muscle regeneration. That's what they call it. Let's see what it says here. Let's see in the bottom. The bottom is always the most important factor. Yeah, muscle cell activation resembles embryonic myogenesis in several ways, including the de novo induction of the myogenic regulator factors. Okay, signaling factors released during the regeneration process have been identified by their functions, remain to be fully defined. In addition, recent evidence supports the possible contribution of adult stem cells to muscle regeneration process, particularly bone marrow. Yes, the mesenchymal cells, which are in the bone marrow, derived and muscle-derived stem cells contribute to new myofiber formation and to the satellite cell pool after injury. The satellite cell pool is important. We want to increase regeneration. The satellite cell pool. They're talking about micro injuries again, dude. That's the regeneration process. Okay, so yeah, relax on that one. I don't know. You get some crazy people out there. Cellular molecular signatures of muscle regeneration, current concepts and controversies in adult myogenesis. These are not important. Yeah, whatever. It's all good stuff, man. So yeah, I put some more stuff there about DOMS, the clicks and the DOMS. So there was that one of DOMS, and then there was this one of DOMS as well. I don't know if you've read this one. Maybe you read it. The myofibrillar skeletal alterations observed in delayed onset muscle soreness caused by what? What is delayed onset muscle soreness caused by? Eccentric exercise. Exercise, get it? Are generally considered to represent what? Damage. They represent damage. By contrast, our recent immuno-something-whatever tests, alterations reflect myofibrillar remodeling, human biopsies, yeah, okay, whatever. It's a lot of stuff here. It too is short as well. It too is short. Our results strongly support that the myofibrillar and skeletal the skeletal of the cytoskeletal alterations considered to be the hallmarks of DOMS reflect an adaptive remodeling of the myofibers. The adaptive remodeling. See, how does your body adapt to getting muscle damage? By remodeling it what? Bigger and stronger, right? And when it adapts to being bigger and stronger, it's now resilient from further assaults, and you're not getting damaged anymore. So you're just a lifting slave in the gym. You're not building myofibrillar growth if you're not bringing back that DOMS and that damage. The DOMS and the damage, because they go together. Okay, friends, I'm gone. I'm out. And then there's another one here, resistance training. You could read this, myofibrillar protein synthesis. It's based on what? Muscle damage. But they're talking about after attenuation of muscle damage. You can read this one too. Physiological, unaccustomed resistance exercise results in what? Muscle damage. Evidenced directly by assessing muscle ultrastructural changes. That's how they did it. Oh, yes, loss of muscle strength, increases in muscle soreness, and creatine kinase. Oh, that's terrible, eh? When you get damaged, there's all those problems. After the initial muscle damage, growth mechanisms are triggered. The growth mechanisms are triggered. How about that, eh? See that? They're not triggered from weightlifting in an adapted state. They're triggered when there's muscle damage. See? When the initial muscle damage, after initial muscle damage, growth mechanisms are triggered. They didn't say after your session of working out in the gym, there were growth factors, growth mechanisms triggered, working out in the gym, adapted to all your weights and your lifting. Producing an increase in protein synthesis to support what? Well, protein synthesis is extended for an entire week. During that, what is it? The tissue repair, the repair and the remodeling. Yeah, that's basically what they're telling you here. Lack of correlation to inertia, resistance, protein synthesis, chronic muscle hypertrophy could be due to damage of protein structures that would require repair and therefore a greater increase in protein synthetic response is necessary. Yes, the larger protein synthesis, you need a lot more. If you get any damage, you need a whole bunch of protein. Now, you working out in the gym, like you work out, you don't need any of that really. You don't need that. You guys are putting just carbs in your muscle. It's just energy you're expelling. You're using up energy, putting back energy because you're lifting slaves. Yeah, it would be result in protein synthetic response being directed towards repair and muscle damage and more toward muscle fiber hypertrophy than, yeah, okay, whatever. So you get the point, man. I don't know what else to say there. That's just another report. Anyways, friends, I gotta go. And you can read some of this stuff too I posted here. You may not understand it. Maybe you will. Myofibrillar hypertrophy refers to when the number of myofibrillars increase. This causes muscles to increase in strength and density. The muscles also contain sarcoplasmic fluid. This fluid is an energy source. See that? That surrounds the myofibrillars and muscles. It's an energy source when you're adapted. Stop eating carbohydrates and you won't load any of that energy source in there. And then your muscles will never, they'll never get jacked. Unless, of course, you build myofibrillars. I don't know. But that just makes you very dense. Sarcoplasmic hypertrophy is greater in the muscles of bodybuilders because studies suggest sarcoplasmic hypertrophy shows a greater increase in what muscle size, while myofibrillar hypertrophy proves to increase overall muscular strength, making it more dominant in what? Olympic weightlifters. How about that? I always wondered why they were able to lift so much. They've always mentioned that. They do different things there all the time. They do, I don't know. They do a bunch of stuff there. Unusual lifts and stuff. I don't know. I can't tell you, man. It just, they say that it's predominant in there. They're just doing a comparison between the bodybuilder and the Olympic lifters. They didn't say power lifters. They said Olympic lifters. The variance, the difference. Okay, the sarcoplasmic to myofibrillar growth. I don't know how much myofibrillar growth Olympic lifters are getting. Do they? I don't know. Maybe they got some. Maybe it's a little bit. Maybe a tiny bit. Some bodybuilders get it too, but they don't get a lot. They get more sarcoplasmic. That's why I try to tell you, bodybuilding it's subjective, bro. It's sarcoplasmic. A lot of phony muscles people are getting in the gym because they don't do Olympic lifting in a regular gym. They just lift weights like bodybuilders. They use machines and all these whatever. Now, as a natural bodybuilder, you can get more myofibrillar growth. You don't have to be Olympic lifter. You just have to do things what it said there. You're unaccustomed to doing, unfamiliar, and untrained muscles get this damage to come back. And then protein synthesis needs to exceed that breakdown in order for that damn thing to grow. Get it? It's really simple. I'll see you in the next one, man. Tell me what you think about that. Like, subscribe, support the channel. See you in the next one. Ciao. Blaha. Yeah, man. I gotta go.